All right, so we'll go ahead and get started. Hello, everybody, and thank you for joining us. Director of Business Development, Dan O'Connor, will introduce our speakers for today's demonstration of the latest version of our flagship PBBM PBPK modeling simulation package, GastroPlus. A few housekeeping notes before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. I'll now pass things over to Dan.
Good morning, good afternoon, good evening, depending on where you are in the world. This is Dan O'Connor. I'm Director of Business Development for GastroPlus. So we welcome you to our 25th anniversary celebration and the next version of GastroPlus, version 9.8.2. Happily, it's my task to describe for you our two presenters and moderators. First, let me talk about Neil Miller, who's our Vice President of Simulation Sciences. He's driving development of application for PBPK modeling, software tools, and running a global team to be able to both increase capabilities and support our customers. Previously, Neil was at GSK for 2 decades. He looks much older than he is, but he had a lot of experience in advancing PBPK, providing mechanistic modeling for drug discovery and development. Neil will act as our moderator at the Q&A session. Our presenter today is Haiying Zhou, who is Director of Simulations Technologies at Simulations Plus. She joined Simulations Plus in 2009. So what is that, 11 years, 12 years ago. And she is focused on the development of modeling software for quantitative understanding of pharmacokinetics and pharmacodynamics of drug candidates, principally focused on GastroPlus. She's also the principal investigator for multiple industry or FDA-funded projects to enhance the current functionality within GastroPlus. So we invite your participation by adding questions as we go, and we'll hopefully have enough time at the conclusion to accommodate those. Let me throw it back to Jasmine for a first poll question. Jasmine?
Yep, so our first poll question is, which application are you currently mainly using GastroPlus for? We'll give everybody a few seconds. Okay, I'll now close the poll, share the results. So it looks like we have kind of a tie between biopharmaceutics and DMPK. And we can now kick things off to Haiying.
Yes, let me show you my screen.
You can see your screen.
Yeah. Can you see my screen? Okay, great. Yeah. Hello, everyone, and welcome to the webinar. Today, I'm going to describe to you many of the new enhancements in GastroPlus version 9.8.2. In this version, we included some new features based on a survey from GastroPlus user group. We also incorporated some of the new functions that we developed from the different collaboration projects. So the first, this slide just to show you a summary of some of the new enhancements in version 9.8.2 in different modules. For the ACAT model, we did incorporate nonlinear biosalt effect, add a version 2 biorelevant medium passive as a default recipe for the biorelevant solubility input. Also improve the suspension dosing. Now with extra user input, the program will set the suspension dosing as a combination of a dissolved solution and also solid particles. Based on the FDA, the food effect guidance, the 2 FDA low-fat meal was also added to the program as an option you could select for your model. For the metabolism and the transporter module, some new built-in enzyme expression level was added, and the ontogeny data function for multiple enzymes was also incorporated. We also improved the automatic fitting for Vmax and Km, so this enzyme activity. This is specifically related with the PBPK model. So when you optimize this enzyme activity, so the different location between PBPK and the gut location will be synchronized. For the population simulation, we improved the workflow for fed and fasted virtual bioequivalence crossover simulation. For the additional dosage route, based on some of the collaboration projects, we did add, for the ocular module, we add a new built-in physiology for 2 different strains of rabbit, and add in the melanin binding to extra location like the sclera and the retina. For the ocular model, we also add a new PD model for the intraocular pressure. For the intramuscular model, we add an option allow user to modify the intramuscular Kp with the compartment model, and also add new built-in mouse intramuscular physiology. For the general features, there are also like multiple items like we allow user to specify the fraction unbound in enterocytes directly in the GastroPlus window, and also like some change with the PSA, and also extra information saved in the simulation output file. So this is some of the, like just some of the points. Definitely there are also some other like small improvements. These are just some of the main items we add to version 9.8.2. First, we talk about the ACAT model. For the ACAT model, for the biosalt effect, if we compare version 9.81 with version 9.8.2, we find for the model parameters, we include one extra. This calls SR exponent. This has an exponent term has added to this function. And we add the passive version 2 medium to for the input, and also allow user to check the fitting. We add the calculated solubility. So you could input the experimental solubility. The program will use this to fit the parameters, and then how good is the fitting, you could check the calculated solubility. So this is some of the change we added to the biosalt effect. Added the passive version 2 as a default recipe. This is because of the in vitro value for the solubilization ratio will be calculated based on this in vitro solubility measured in passive or passive or some other like biorelevant medium. So we only add the version 2 for the passive. This is because the passive version 1 and the version 2 have the same exact biosalt concentration, and that will be a redundant input for if we include both, because the pH and the biosalt concentration are the same. So that's why so we only include the version 2 passive as a default excipient. So we also included the exponent term, so we call it N. So with these parameters, we could incorporate the nonlinear biosalt effect. So without this, and with previous versions, so that model is like a linear biosalt effect. If we look at the equations, and this is how the model link the biosalt concentration and with the solubility at the specified biosalt concentration and the pH. If without this N, so this is like a linear equation. So this is so if with this extra exponent term, so we could have like, especially if this N is not equal 1, this more represents a nonlinear biosalt effect. So if we look, you could also check whether you want to include this in the fit. If you include this in the fit, the program will use your input biorelevant solubility to fit both solubilization ratio and also this exponent term. Since now you are fitting 2 parameters, so the model will check, make sure you have at least 2 biorelevant solubility input. Otherwise, you could only fit just 1 parameter. If you set this exponent value equal 1, so then the program will be, this will be a linear model. So this slide just to show you like the example how this exponent function for the linear relationship and for the nonlinear relationship. After we incorporate a mol file, a structure file, so like for midazolam mol file, the program will predict what's the biorelevant solubility. This experimental solubility actually for this case, this is in silico predicted value. And the model during the import structure process will automatically fit it to a linear model. So this solubilization ratio is fitted with the exponent equal 1. So we could also find with just a linear model, the calculated solubility are very close, like are the same compared to the experimental solubility. But how about if we include this n exponent term in the fitting, then the program, if we include this n to in fit, and then we click on fit to in vitro data, the program will fit to both these 2 parameters. We could find this exponent term are very equal, close to 1. And the solubilization ratio, this value are also very close between the linear model and also the nonlinear model. If you look at the calculated solubility, also these are the same as the experimental solubility. So with extra parameters, so because this is a linear relationship, so with extra parameters won't like play significant role. But there are some cases like for the loratadine example, after the structure import, we do have like the encyclical predicted solubility. If we use the linear model to fit the solubilization ratio, and you could find the calculated solubility do have some difference, especially under the in the passive medium. So if we include this exponent term in the fitting, you will find, so this is more fit to a nonlinear relationship. So for this exponent term, this is 1.7. So it's significant difference compared to 1. And also the solubilization ratio, you could find there are also like a significant difference between this linear model and also the nonlinear model fit. And if we look at the calculated solubility, and for this specified biorelevant medium, and we could find the value are very close to the experimental data. So we just like with this function, we do provide the, like if you could include the extra model parameters to improve the fitting. And also the program will show you like you could have the opportunity to check the fitting result by comparing the experiment solubility and also the calculated solubility. How this will be affect the solubility in our GI tract. So we also compare with the loratadine. We compare under fasted and fed condition with the specified biosalt concentration and the pH at the different GI tract location. And this blue line shows if we use a linear model, the linear fitted model, this is the solubility at the different segment of the GI tract. So this orange line shows with the nonlinear model and what will be the calculated solubility at the different location with the different biosalt concentration in the GI tract. And for the fed condition, so the biosalt concentration now is higher at the different locations. The blue line also shows the calculated solubility, and with the linear model, and the orange line shows the calculated solubility with the nonlinear model. And with this nonlinear model, and we could find the calculated solubility, and both fasted and fed conditions are lower than the linear model. How this will affect our simulation? So in our simulated the plasma concentration versus the time profile. So we also could find under fasted condition and the fed condition. So the simulation with the linear model, they shows like a higher Cmax, and also the whole the line is higher than the nonlinear model. This is because for this compound, the solubility is this is a limitation. So the dissolution is a really limited step. And with a lower solubility with a nonlinear model, this is limited the absorption to the system and also appear in the systemic circulation. So in all these models, the distribution and also the clearance are calculated based on the in silico model. So you may find that if your model is different compared with CP profile, because now all of these are just using in silico prediction. Another a significant improvement is about the suspension dosing. So with version 9.8.1, if you selected a suspension dosing, all the dose will be assumed is undissolved, it's in the solid particles. In some of the simulation of the studies, we did find like for the suspension, we need to consider some amount has been pre-dissolved in the formulation. And in that, in version 9.8.1, the user need to incorporate the mixed multiple dose. With the mixed multiple dose, you could set part of the dose as a solution, and another part of the dose as undissolved as IR suspension. So this incorporates like extra steps. You do able to run the simulation, but it incorporates extra steps to set the dose. So with version 9.8.2, if you like select IR suspension and you have an extra like button name like suspension solubility, you could set some extra like inputs with this formulation. Because the suspension dose now we consider it in this new version as a combination of dissolved solution and also the solid particles based on the vehicle solubility. And also for the oral dose, the suspension volume is in addition to the regular dose volume. So it's like a glass of water. And so this suspension volume needs to be defined in dose volume per dose. So in that way, we could incorporate like the suspension like in the mixed multiple dose or with the different dose level. So the program will able to calculate what will be the suspension volume. And for other dosage routes, normally we do not take like extra fluid like to dilute it. So then the dose volume will determine what will be also the vehicle or the suspension volume. So since there are two extra inputs we need, one is the vehicle solubility, and also another one is the dose volume, the dose volume per dose. So if you click on the suspension solubility, so there will be a form window pops up. You could have the extra suspension vehicle solubility setting. You could set what will be the solubility in the dose vehicle and what will be the vehicle volume per dose. So with this, and then the initial dose, the program could calculate what will be the vehicle volume. And if you have like the solubility in vehicle is 0, that means the program will treat all the dose in all the dose as a solid undissolved form. So based on these two parameters, the program will calculate the amount as dissolved and the amount in solid. For this one, so the calculated is the percent of dose in the solid as the undissolved form is 50% of your total dose. We also add a function like you could estimate the solubility in your vehicle, assume there is no surfactant. Like if you double click on the solubility in those vehicle, and the program will pop up a window, and you could set what's the pH in your vehicle, and then the program will estimate like the solubility in the vehicle. But if you do have surfactant, that means you need to provide this value. So if this is like a different, the pH has some difference, you do able to use this double click function to estimate this solubility. So for this example, we could say like the percent has been dissolved if there is no surfactant. It's very, very small, almost 99%, 99.97% is still as the solid undissolved form. How this will affect our simulation? So if we allow the program try to like based on this formulation information to calculate the dissolved amount and also the undissolved amount. For this example, so we have like based on this input, the percent of dose as dissolved is 5%, and the percent of dose in solid is 95%. And for this compound, the orange line shows like we do consider the 5% as dissolved, the simulation, and this is the blue line shows if it's 100%, we all consider as a solid form undissolved. We could find like even though just for 5% dissolved, this shows significant difference in the simulated Cmax and also the Tmax. So they do show, because for this compound, they do have very low like the solubility. Also, you could incorporate this suspension like the formulation parameters for PSA. So for this one, you could include like the solubility in the vehicle and and also the suspension volume per dose. And for the PSA, I just run a 3D PSA simulation, but I plot it as a 2D. You could find as the solubility in the vehicle increase, and the Cmax also increase. And the different lines shows the different volume of the suspension per dose. So that means if you have like the higher like the volume, and also you will have more get dissolved, you will have more like the higher Cmax. Yeah. For other dosage routes, we also incorporate this function for this improved suspension dosing. But for other dosage route, we will treat the dose volume as a suspension volume. So this example shows for the ocular topical suspension, the initial dose as 5 milligram and the dose volume is 0.1 milliliter. And with this one, for the suspension solubility, suspension vehicle solubility setting, you're only able to change the solubility in the vehicle, because for the suspension volume, we assume it's the same as dose volume. So we do not want to give you like two location for this input that cause confusion. So for the other dosage route, the suspension volume will be the same as the dose volume. For the another is in the improvement in the ACAT model. So we provided extra FDA low-fat meal options. These two meal has been described in the food effect guidance. And one is a low-fat 450 kcal, another one is low-fat 387 kcal. And in both of these meals, the percent of fat in the meal is close to 25%. One is 25%, another one is calculated as 23%. So with you selected this different meal type, the program will based on the meal type, meal calorie, percent of fat in the meal to calculate what will be the stomach transit time and what will be the bile salt concentration in the GI tract. So this will be all handled automatically by the program based on the meal input. For the metabolism and the transporter module, we did extensive literature research to try to incorporate some of the new enzyme expression. And with this slide just shows this is in kidney for the version 9.8.1. So in version 9.8.2, we do add CES2 and also different GST enzyme in the program as a default physiology. If you create a PBPK model, all these different expression will be incorporated in your PBPK model. And also add some new ontogeny function for different enzymes. So in this slide, we just show you an example like CES1. So you could say for the 20 weeks pregnancy, the expression level in the fetus, and also the 1 year old female in the liver, and also 30 years old female the liver, the CES1 expression. And we can find like during the pregnancy, the expression level in the fetus like continue like increase the expression, and also even after the birth, they still like continue like increase the expression level until reach the adult level. And also you could also find like in this table, because just the space we couldn't show all the enzymes. There are also some like the different enzymes, their expression change with age. And also you can find for GSTP enzyme, it do increase the expression like during the pregnancy, and after 1 year old, the expression level is already like the same as the adult level. So the ontogeny, like the function for all the different enzymes, has been incorporated in the program. For some of the enzymes, we also find that it might have like the gender difference. So we also add like a built-in gender difference effect in the enzyme expression. For the UGT2B17, we could find for this 30 years old female, and the expression level compared with a 30 years old male, and their expression like for the female is significantly lower, and the male is 2 times higher. So if the program find for the program like it has the gender difference, then if you create an individual subject or do population simulation, so all this gender difference will be incorporated when generate the subject for the proper like the enzyme expression. For the another improvement in the enzyme and the transporter module is if you try to optimize the Vmax and the Km, if this is a PBPK model, the program will provide you an extra like input. If you check this checkbox, then that means so you want the program to synchronize the enzyme fit at the GAT location and at the PBPK location, because you don't want to optimize them separately. These things if they are the same enzyme, like for the in this example, like for this 3A4. So the 3A4 enzyme activity in the PBPK model location and also in the GAT location, after the optimization, the Km will be the same and the Vmax will be synchronized, taking unit into account. Like in the GAT location will be milligram per second. In the PBPK location, it will be milligram per second per milligram enzyme. So you do not need to worry about, so I optimize my PBPK location, the enzyme activity. I need to manually convert it to find what will be my GAT like Vmax and the Km. Now the program will automatically update this, Vmax and the Km. Another improvement is related with the population simulation, especially about the fed and fasted virtual trial, like for this bioequivalence crossover simulation. So the program will be automatically detect if this is a fed or fasted crossover population simulation. If they find this is like some difference, like in this crossover for the fed condition and the fasted condition, the program will automatically hold most of the physiology parameters fixed, because these are described by from like the population simulation result you load in for most some of the physiology parameters related with that subject, except for parameters that will be changed in the fed versus fasted condition. Something like for the same subject, their fasted and fed condition, the stomach pH, stomach fluid volume, stomach transient time, bile salt concentration, and the liver blood flow will be different. Then the program will automatically take care of this, but other parameters will be fixed based on the file you load to the program. So in the previous version of GastroPlus, the population simulation engine would automatically fix all the subject related like physiology parameters So when a previous population result was loaded for a crossover, if you want to simulate it, like use this population simulation result, but run a different state, like you load a fasted condition, you want to run a fed state like a crossover study, then the user need to manually go back to this result file, like STC file, manually edit these parameters. So that means it's stomach pH, stomach fluid, all these parameters listed above. So to facilitate these new options, with this new version for the STC file, they will also save what will be the animal species and also what was the prendial state. And if this you load simulation result, like population simulation result is created in previous version, and we also have the feature in the program automatically detect the fasted fed state. So that means like even though you have a population result from previous versions, the program will also be able to handle it. So this is a new like result from the population simulation. It's added the prendial state. So this is a fed condition. The animal species is human. So that means if you load this file like a fed like result file for a fasted simulation, then the program will pop up a warning for you. So the current crossover like trial is set up to compare fed and fasted state. And if you click on yes, the program will automatically like adjusted the physiology related like to the bile salt concentration, the stomach pH, all these physiologic parameters that will be related with fed and fasted conditions. If you click on no, so that means all the physiologic parameters will be fixed, and this will be user value read from your the result file load in for the crossover. If you really want to modify it, you need to go to the file, STC file, modify it. For the enterocyte fraction unbound, in version 9.8.1, you need to go to the drug table and also find like the fraction unbound in enterocyte to change it. This add some extra like a step if you want to use modify these parameters in your simulation. With this new version 9.8.2, so you could modify these parameters under this gut physiology. So there will be an extra term for the fraction unbound, and you could change these parameters. So some of the we'll question like how will this affect your simulation if you use different fraction unbound in enterocyte. For this simulation, we use midazolam as an example. So the blue line shows there is no binding in the enterocyte, and the orange line shows there is 99% is bound, only 1% is unbound. You could find like there is like a decrease in Cmax, and also because of the binding in the enterocyte, it will slow release, and then you will also find a longer Tmax in your simulation result. For the ocular, like OCAT physiology, we did have like for this module have a significant improvement based on one of the FDA-funded collaboration project. For this ocular physiology, we add 3 more physiologies. One is updated human physiology. For this human, we have a new human version 2021 version. And we have 2 strains of rabbit. One is Dutch belted rabbit, another one is New Zealand rabbit. And within this project, we have scientists did review all the published paper to find all the physiology parameters for the human ocular physiology, and we updated this. And you could find like with the older like the physiology, we name it, still keep the name as a human, but for the updated one, we use the human version 2021. And so the multiple compartment has been changed based on this new literature review. And for this example, I show you in ICB, and you could find the volume for this ICB has been significantly increased from 0.02 milliliter to 0.06 milliliter based on the latest literature review. And also created two new rabbit physiology. One is NZ, the New Zealand rabbit. One is the Dutch belted, like we call it DB rabbit. And in our previous version of GastroPlus, for this default rabbit, we didn't separate like which strain of this rabbit is. So this it's a general rabbit. We didn't know which species. We didn't account the species difference. And now we separate the two like specific species. And for this ICB, like the compartment, you could find there are significant difference in the volume and tissue volume and also the melanin content. Like for this Dutch belted rabbit, it has a significant higher volume and also melanin content, because for this rabbit, their eyes is much bigger compared to like the New Zealand rabbit. So the different locations, I just show you just one example in this ICB, but there will be at the different location, all this physiology will be different based on the species difference. For this validation examples, we try to run some of the simulation and with the different physiology. And in this example, I show you like with the different rabbit, NZ, New Zealand rabbit, and the Dutch rabbit at some of the locations which have the experiment data. One is in the corneal, another is aqueous humor. And by like a first glance, you may find the results are very similar. But if we consider like in this study, they use the New Zealand rabbit, and we could find the New Zealand rabbit like especially for the aqueous humor, like the model prediction is better. And if we look closely, like for this aqueous humor and ICB result, and this blue line shows the baseline, like with the default rabbit, and the red line shows the New Zealand rabbit, and the green shows the Dutch belted rabbit. And we could find like after we consider the species difference, and then the dynamic of the concentration profile in the aqueous humor got improvement. So they have a shorter Tmax. And also the significant difference in the ICB simulated the concentration profile. And so with the consider the species, specific species, the concentration has been decreased. Also, we add melanin binding based on the literature research to extra location, like in the sclera and also in the retina. At these locations, you could find with this new version, they have the extra term like for this melanin like content. So to validate this like melanin binding in the effect in the local concentration, so I did show a validation result. The simulation shows like for with and without melanin binding in sclera. With the binding, we set the data to 85%, which is similar to the parameters fitted to ICB. So in this study, the ICB, they do have observed data. So we could find like without melanin binding at the different segments of this sclera, so the model prediction underpredicted the profile. Then if we consider the binding, both of the simulated result at the different location got improved. So that means the addition of this new physiology mechanism in the sclera significantly enhances the model prediction in the sclera. For this ocular model, we also add a PD model. So this is intraocular pressure model. So this is, we incorporated the multiple model structures. If you have the access to this PD plus module, and you will find we added two more model and as a direct model, so model 1, model 2, and also two model in the indirect model. So the difference between these two models is indirect model, we add extra delay, so described by the transient through this subcompartment. And also the model 1, model 2 represent one is inhibit the production of the intraocular pressure, and when is stimulated, the disappear of the pressure. And also the baseline, like the IOP, this pressure represent a Cascadia rhythm in both rabbit and also in human patient. So to account this Cascadia rhythm, we also include this function in this pressure production, IOP production. Furthermore, the different species shows different baseline, like the pressure. For the rabbit model with the different strains, so we find that there is no significant difference in the observed, like the IOP, like the data. So that means at that time we do not have any drug effect, but there like IOP, like the profile is very similar between the different species. So this 0 means the midnight. So that means like from midnight and how the pressure change within 1 day. So this is the baseline IOP model. So we need to incorporate also before we start the drug, the drug therapeutic effect. We need to consider the baseline model. And this is also the collected data to derive for the monkey IOP model. For the human, so this will be 3 types of different baseline model. One is they call, from the literature, they call it no rhythm. So this is account 20% in the subjects. And another one is coterminal rhythm. So this is account 40% of the subject. And another one is diurnal rhythm. So for this one, in the measured subject, it's account 50%. And so you could find like how it's changed within 1 day. So their profile are different. Also, we need to consider for the glaucoma patient, and also they have a different 2 types of like different disease for this glaucoma. One is still have the normal tension. Another one is has a hypertension. So that means, so in your model, when you select the baseline model, so if it's a human, so then you're able to select what's the disease status, whether it's normal intention like glaucoma or it is hypertension glaucoma. Also for the rhythm, whether it's non-rhythm or it's like nocturnal or it's diurnal. So all this like you could select. So totally the program based on your selection will provide you 6 sets of baseline of parameters for this IOP model. And then based on your compound, you will focus on this IMAX or Emax model, only describe your compound therapeutic effect. Another one is for the improvement is in the intramuscular. For this intramuscular model, we allow you to give the Kp input for this compartment PK model. So in previous versions, the program estimated for if it's a compartment model, and the program will estimate the Kp at the injection site intramuscular location based on some default tissue composition, muscle tissue composition, and the user couldn't modify it. That caused some problem if user do have measured like the muscle Kp, and also if they want to optimize these parameters, they try to obtain better prediction like for the tissue concentration or the PK profile. So within this new version, we do provide this as an input in the muscle, like intramuscular module window, and you could provide it. So initially the model shows to you is a model prediction based on some default muscle composition. And this use like the Lukasheva method and calculated this Kp. So provided for you. You are able to modify it if you have measurement data, or if you want to optimize it. And if it's for PBPK model, the program still show you the Kb for the intramuscular location, but you couldn't modify it because this value will be taken from the PBPK summary table. We do not expect at the injection site muscle will have significant difference in the tissue composition compared to the PBPK muscle tissue. So that's why these 2 values will be consistent. If you really want to modify the intramuscular, like the Kp value, you could change it under the PBPK summary table, and these two will be synchronized and take the value from the PBPK summary table. So another one, it is for the intramuscular physiology. We also add a mouse sign, like the location for this physiology, like it will have a different blood flow compared to other locations. Now for this intramuscular, like the physiology, so the available species are human, dog, monkey, rat, and mouse. So we do add a new, like the mouse physiology. So I think these are some of the new features I want to describe today. And before I take any questions, first I want to acknowledge multiple scientists from our company, like Jim, Jessica, Manash from the simulation technology group, Maxim from the regulatory fair teams, and also Norm from the simulation study group. And also thanks Neil, our vice president, and Michael Bolger, our founding scientist, and Vera Lukachiva as our chief scientist, their extra support and also suggestions. Also other teams at the Simulations Plus also provided lots of support. The chemoinformatics group, the computational group, the research fellows, and also the simulation study group. So I think Jasmine has another poll. After that, we will take any questions you have.
Yes, so our next poll is, what's your experience with PBPK modeling? And right after this, we'll go to questions. We'll give everybody a few more seconds here. And it looks like we're kind of everywhere, but that's good to know. All right, and we'll pass things off to Neil for the Q&A.
Thank you, Jasmine, and thank you, Haiying, for a very thorough overview of all the enhancements in 9.8.2. So we do have a number of questions, and we will take those in the order that they came in. So the first question is related to aqueous solubility. So which aqueous solubility do you use when different biorelevant solubilities with different pH are used as an input?
So this aqueous solubility will be based on what you input in the program as the reference solubility, and also the pKa it described it. So the program has a built-in pKa model, like based model, and then the program will derive based on the reference solubility and also the pKa derived at the different pH. This will be the aqueous solubility it will be used. And then, so if you have a biosalt, this will be incorporated in the biosalt the solubility calculation. So yeah, as I mentioned, for summary, for this aqueous solubility, it will be calculated based on your reference solubility and your pKa's. So they will use the built-in pKa relationship to calculate the solubility, aqueous solubility at the different pH.
Thanks, Haiying. So if you enter some biorelevant solubilities at a pH of 5 and 6.5, the software will calculate what the aqueous solubility is at those pHs, so that it then can account for the correct bile salt enhancement. The next question is that someone said, my reconstituted vehicle pH is around 7, but my suspension mostly absorbs in the stomach. I have dissolution release data in pH 1.2 and 2.1. For a better simulation, what should I consider, either the drug release or the reconstituted drug solubility?
Okay, let me okay, I think for this one, so you did have first so that means your suspension did get dissolved in the stomach at the pH equal 0.1 and 2.1. So that means you still have like just give the program the information of your formulation. So your vehicle pH is 7, and then what will be the like before you dose it? What will be the predefined like dissolved amount, and also the amount in solid. But when it's dosed in the stomach, and because of the different pH, your stomach will get dissolved, and at this lower pH, the program will consider the pH difference. This will be automatically calculated. And the problem is you do mention like it's get mostly absorbed in the stomach. So, but within the GastroPlus currently, like for the ASF model, we consider the absorption, like the permeability at the different location of the GI tract. So if you like have a absorption in the stomach, that means you need to add a stomach like ASF, manually add it to account the absorption. But to consider the like the dissolve of your formulation, so you could like just put in your vehicle pH like at 7 to calculate the dissolved amount in the formulation. The program will calculate how your formulation get to the stomach, get dissolved based on the pH difference.
Thank you, Haiying. So the next question is related to the synchronization of the Km and the Vmax. So that feature, the question is that feature applied also for transporters as well as enzymes?
Yeah, so currently we like put it in enzyme, because in enzyme we do link the like the expressions like in the stomach, in the GI tract as like relevant percent as what the expression in liver. But for the transporter, it could be different, because for the transporter, for the especially for some default expression level transporter, we reference it to a segment of the GI tract. So the expression level, like in the GI tract and also the tissue, are not like linked. I would say are not linked through the liver, like the expression level. So that's why currently we work it with the enzyme. For transporter, if we want to do that, then we need to have the information how to link the expression difference between the gut location and also the PBPK location. So for the enzyme, we linked it by relevant like the gut location expression level to the total liver expression level. So we have a way to link consider the expression level. We could take out the expression difference, and then we just consider the Vmax and the Km. So for the transporter, because the expression at the different location of the GI tract are not referenced to the liver expression. They are referenced to one of the segments of the GI tract, like some with relevant might to colon. So that's why currently it works for the enzyme.
Thank you, Haiying. The questions are coming in thick and fast now. This might be a record for the number of questions. We'll try to get to all of them, but if not, we'll follow up with the individuals. The next question is, are the literature references available that support the new parameter data so that they can be used to demonstrate scientific support for parameter values as part of support for the PBPK results? So are the literature references available?
Yes. So the extra literature will be available like in the manual. And also for some, like we do add extra, like for the enzyme expression, for some related with the enzyme, we do also like if you click on this help button, it will show you like for these different parameters, what this new literature like reference will be added. Yes, it will be added in this version. Yeah, in different version. Yeah, if we add the more literature, we will add like to each like version. Yes.
The next question is to do with virtual bioequivalence assessments. And the question is, can partial or full replicate crossover designs be simulated in virtual bioequivalence assessments?
So is this question about like you only select partial of the subject from for a crossover study?
I think so, yes.
Yeah, I would say currently the function, so we only allow like the full replicate crossover. but you do have some functions like when you run a population simulation, you could have like a small group, like repeated, like the crossover, repeated population simulation. Then when you do crossover, you do not need like if you run like a population simulation for like 10 group, and each group you have 25 subjects. So for the crossover, it doesn't mean you have to run like these 10 groups, like 25 subjects, like 250 subjects. You could select any like subgroups you want to do the crossover design. So that means, so yeah, you could do like the partial like crossover, but that means you are not select all the subjects, because the subject will be because it's repeated like a population simulation. You run 10 group, you could based on this group to select them for the population, like the crossover study. Yes.
Okay, thank you. We've had another comment on that one. And if full replicant means that you do the test twice and the reference twice, and partial means you only do them once, then that can be done with the software.
Oh, excuse me, I missed that question. What's that?
So if the question was around, is the full replicate meaning that you've done the test twice and the reference twice, and the partial means that you only want to do them once, then you can do that with the software.
You do partial is once, and then the full you do it twice.
Yeah, if you don't want to repeat the whole of the trial and just want to do partial of it. It's not a question. That's just that's in elaborating on that answer. If the question that someone has posed is, do you have to run all of it? So do you have to do it twice? No, you don't. You can do it once rather than running it twice.
Yes, you can do it. Yeah, yes.
Okay, we've got a few minutes left. Let's have a look at the next question. Is how do you decide on the % unbound for the enterocytes? Is it based on fitting, and does it have any relationship to lysosomal trapping of the compound? I guess I could answer that one to save you putting you on the spot all of the time, Haiying. Is that we do have another software product, Membrane Plus, which we can use to give us an estimate of the fraction unbound in the enterocytes. You can also fit that data as well. So there are a number of ways that you can come up with that fraction unbound in enterocytes. If you have a BCS class 1 compound and you're expecting fast absorption and you're not seeing it, then you can investigate now having that fraction unbound in the enterocytes exposed on the GUI on the physiology tab. You can easily investigate the sensitivity of your simulation to that parameter. The next question is asking us to go to slide 35 and is saying, is there human quadriceps physiology available?
Yeah, it seems like we have like the three, like the building physiology. We do not have this like quadriceps. Yeah, so we could check like whether we have the data, published data, then we could add it. Yeah, so currently it seems, yeah, we have the other three type of muscle.
Okay,
but
the next one.
Yeah, but you do able, this is the default physiology, but if you do know like the different blood flow or this physiology, like for this human quadriceps, you could add it like by yourself. You could like select one of the human and also you save as it and then modify the value. So you could like also build it by yourself. Yeah.
Okay, we've still got about half a dozen questions, so we're not going to get to them all. I'll pick out one question to finish with, but I'm just going to, this one isn't a question. This is just, someone said it's an extremely interesting presentation, lots of improvements in 9.8.2 and a well done from one of our users. So thank you very much for that. Let's just pick out one of these questions that we've got left here. Okay, so what is the appropriate situation to use the Moody model on the physiology tab in the simulation? So this isn't a specific enhancement at 9.8.2. It's been in the software for a while. So this is having a dynamic fluid volume model as part of your absorption and compartmental transit model. So what is the appropriate situation to use the Moody model?
Yeah, so
I guess in my experience, I think it's used as an exploratory kind of tool in our toolbox to see what the sensitivity is for your particular compound. We haven't got data to parameterize dynamic fluid volume models in all of the species for all of the different prandial states. So therefore, if your compound is sensitive to the fluid, it's solubility limited, I would use it to explore and to see would be my suggestion.
Yes, I think that's true. Yeah, if it's like you could do some testing like before you use Moody model. Like if you find like you said the different like the fluid in your small intestinal, will this very sensitive parameters will affect the simulation? Then you might want to use the Moody model, because that means like the glass of water you take in, how it gets absorbed will affect like the upper small intestinal, the fluid like content. Yes, this will be affect for such a compound. It might be affect your simulation. So definitely, I think you will want to like so you want to do something like whether your compound is a low permeability, and how will this be sensitive related with the available fluid for dissolved. Yeah, then you will consider to use the Moody model. And also there are like a different type, like for this absorption like rate you want to use. So currently we do not have a suggestion which will be the best Moody model, but you could select a different model to explore which one could best fit your simulation.
Thank you, Haiying, and thank you once again for a very interesting and detailed presentation, and thank you for answering those questions. Apologies to those people who we haven't got to your question, but we are at time now, and a big thank you for spending the last hour with us today. Hopefully you're excited about getting your hands on version 9.8.2 of GastroPlus, and we look forward to speaking to you again soon. Thank you, and take care.
Thank you all. We invite our audience to learn more about GastroPlus on our website. You can find some of our team at this year's AAPS meeting. And I wanted to let you guys know that this webinar has been recorded for playback, so it'll be available on our website and YouTube. Thank you again to everybody. This concludes our webinar for today. Thank you.